Welcome to the GGA seminar. I am hosting today's speaker. I'm super excited to introduce you to Alex Dornberg from UNC Charlotte. Just a little bit of a bio background about Alex. He got his bachelor's degree in zoology from Washington State University. He then moved from the West Coast to the East Coast, where he got his master's and PhD in ecology and evolutionary biology from Yale University. After he got his degree, he did a brief postdoc at Yale before he moved to the thriving metropolis of Raleigh, North Carolina, where he was research curator of ichthyology at the North Carolina Museum of Natural Sciences from 2015 to 2020. And it was during that time that I first met Alex. It was 2016. He gave a seminar out of the vet school on ice fishes. Now, before that time, up to that time, I was very hyper-focused on using zebrafish as an animal model, but I was struggling to get NSF grants because I had reviewers saying, well, that'll tell us a lot about zebrafish, but what does it tell us about any other animal species? So I was very excited to meet Alex. We had great conversation afterwards. We started having coffees together. And in 2018, we had our first grant funded by NSF, focused on the evolution of immune receptor genes across multiple fish species, getting into more ancient fish species. So that collaboration really, I believe, helped me move my career forward, helped move Alex's career forward. And I use that as an example for the graduate students and postdocs, how important it is to go to seminars, hear other people speak, because you never know, you're going to hear a topic or you're going to meet a speaker who's just doing the coolest thing, and then it intersects with what you're interested in. And, so I like using that example when I'm talking to trainees, because go to conferences, go to seminars, and hopefully someday you'll meet your Alex Dornberg. So that was awkward. In 2020, Alex sadly moved away from the area. He moved to Charlotte. He became accepted a position as assistant professor in the Department of Bioinformatics and Genomics at UNC Charlotte. And over the years, the more recent history, he's had funding from National Science Foundation, a couple collaborative grants with me looking at immune genes. He's also had grants on computational epidemiology and evolution of SARS and COVID-19. So those are things I've never heard Alex talk about. So I asked him if he could come and not talk about the stuff I already know about, but the stuff I haven't heard about. So that's the title today, is Forecasting Reinfection Risks with Evolutionary Medicine. So with that, I'm going to hand it over to Alex. Welcome.
Thanks for the really nice introduction. I'm not going to guarantee funding to everybody, but I am really excited to talk to you. I've really enjoyed actually talking to everybody just up until right now. It's been great. Everybody here is really nice, super enthusiastic, and I'm going to try to focus, but there's been so many cool ideas that I am scatterbrained. But with that, I'm Alex. I'm over, I don't know how to use this pointer, so I'm just going to chase, or not. I don't know what I'm doing. Maybe?
Pointed at the screen.
Well, you have to hit a button on the keyboard.
You might need to click on the
green dot up there.
Oh, is
with
it?
the keyboard
Okay, that's working.
to click on the slides themselves once to refocus.
Mouse.
There we go. I can use the mouse and now hurt me.
There should be a video here. We'll display it this way.
All right, how many bioinformaticians does it take to start a video? I don't know. But just to tell you about myself, I run the Evolutionary Informatics Lab at UNC Charlotte. So basically, we're interested in that intersection between informatics, evolutionary biology, and genomics. And I mostly work on these things. I love rayfin fishes, ignore the shark swimming in the background. But rayfin fishes are, to me, just really fascinating, really, oh, it is being transcribed. And it's about half of all living vertebrates. And we've come a really long way in understanding the evolutionary history of these things. Like in the last 10 years, our ability to resolve the rayfin fish tree of life has just gotten better and better and better. And in fact, thanks in part to Lily, who's done some really cool work on this frontier. And with these trees now, we're able to map all sorts of cool comparative data. So a lot of my research is also focused on mapping things onto the rayfin fish tree of life for the lineages that live in Antarctica. There's a really interesting group of fishes there that Jeff mentioned called notophinioids, or Antarctic ice fishes, which I won't tell you much more about today, other than they're really bizarre at the genetic level. So a lot of the traits that they have are disease states in us. So Some of these lineages don't have hemoglobin in the blood. Some of these lineages co-opt genes that are linked to skeletal disorders in humans. They use those to achieve buoyancy. They have bizarre kidney functions. So it's a really cool group of organisms. I also spent a lot of time working on coral reef fishes. I talked to some of you about this. So I'm kind of interested in life in coral reefs. And lately, I've also been very interested in sort of evolutionary transitions and the genetic changes that go along with these. So we have a lot of projects going on with mudskippers and other lineages to look at immune gene evolution. So some stuff I'm working on with Jeff as well. And then lastly, we've done a lot of work together collaboratively here in North Carolina, working on these things. These are gar, the sister lineage of the bowfins. These are things Darwin referred to these as living fossils, and they're really wild at the genetic level. They have loci in their genome that look more like those in ours than those in other ray fin fishes. So if you're interested in any of this, I'm happy to tell you about it. But when I moved to Charlotte, it was right when this started happening. I started a job I wasn't allowed to go to, which was like the most surreal experience ever. But being, by the way, I'll tell you guys about that some other time. But one of the craziest things that probably happened to a lot of you was like, we're in this major international crisis, trained to do all this stuff. So the question became, what can we do to be useful? And that's sort of how I launched into working on coronaviruses. Before kind of jumping ship and talking about this wholesale, one other thing I'm interested in, if anybody else wants to talk to me about it, I'm also really interested in education research and doing a lot of things relating to education and language and artificial intelligence. So if that's something you want to talk to me more about, I didn't make a slide on it, but since I've been talking to people about it, I thought I'd tell you about that. So kind of prior to the pandemic, when I thought about impending biodiversity crises, things of that nature, a lot of my research was sort of focused on this impending storm, right, of humanity is just developing and developing and developing. How are lineages going to respond to rapid environmental change? And one thing that I sort of started to transition towards working with Jeff and thinking about more and more was in the backdrop of all this, there's this kind of constant specter of disease, right? So lineages through the history of life on our planet, right? Pathogens have shaped evolution in fundamental ways, right? And the more we start looking at like the impact of parasites, the impact of infections, so on and so forth, right? We can see, wow, there's like tremendous changes in the way that lineages are able to achieve evolutionary transitions. There's all sorts of stuff that pathogens have done to shape the history of life on our planet. And thinking about the history of pathogen exposure in human history, there's also this history of plagues and pandemics that we've been documenting for thousands of years, starting back with the plague of Justinian's, Japanese smallpox, and bubonic plague, plagues of the 17th and 18th century. Got yellow fever, Spanish flu of 1819 or 1918, Russian flu, Hong Kong flu, and Asian flu, and then up to the present day, right? We're still sort of in SARS-CoV-2 hasn't gone away; it's still here. And kind of thinking about the toll this has taken, right? You start with like 200 million people died of bubonic plague. We lost 40 to 50 million people to the Spanish flu. Zooming in, this is an older slide, right? But over 5.5 million people died to SARS-CoV-2, right? So it hasn't gone away. The thing we like to pretend it's not around as much, but hospitalization due to COVID-19 remains 3 times higher through time than hospitalization due to influenza. So here's April 2021 going through to October of last year. Here's weekly hospitalization rates of SARS-CoV-2 versus hospitalization rates of influenza. You might be like, Alex, this is old data. Surely it's not that bad now. Oh, it's just as bad. So here's the rate of hospitalization per 100,000 people. That took this off the CDC website yesterday to make sure it was up to date with what the CDC is reporting. Here's the rate of hospitalization of COVID-19. Here are rates of hospitalization for flu and RSV. And so COVID remains this scourge to public health. And one thing that started thinking about more and more is outside we have all these pathogens, right, that are constantly trying to invade our bodies. And as the pandemic started, along with a lot of other people, were like, well, what do we have in place to prepare us for pathogens? And do you guys know when? Thank you for. I didn't know how to get rid of that bar. It's been driving me crazy. Do you guys know when the United States first started to officially draft pandemic response policy? Shout out some.
So you mean for like overall in the United States or for specific like COVID-19 like virus?
So for a pandemic, like preparedness for the next plague policy, like if yeah.
1806?
What's that?
1806?
1806. We'll play by Price is Right rules. This is the interactive portion.
Some of us accidentally got a sneak peek.
Was it you who didn't cheat? At
2000.
2000? Okay, so we're somewhere between the 1800s and 2000s. So we officially started drafting around 1976. This is what your Apple computer would have looked like. It's also the year that megamouth sharks were officially discovered by science. Pretty cool species. It was about 2 years later that the first official policy was drafted, but this was prompted by a swine flu outbreak at a military base in New Jersey. So there's this outbreak at Fort Dix, about 200 some people got sick, one person died, and there's still memory of the 1918 flu. There's still memory of several other influenza pandemics that have swept through and killed millions of people. So rightly so, a lot of people were absolutely terrified that there's going to be another large outbreak. And so this led to some really interesting parallels. So Gerald Ford decided to vaccinate the entire US population at warp speed. I don't know if this sounds familiar to anybody in your recent history. And this actually ended up being quite the public relations disaster. I'm not going to go into too much detail here. It's a really interesting piece of history with ramifications all the way through the present day that somehow nobody except the BBC even mentioned during the pandemic, when you were literally seeing the same news headlines that you could have transposed from 1976 to 2020 concerning vaccination. But anyway, if you guys want interesting reading, look up the swine flu fiasco of 1976, some really interesting history. And during this time, Gerald Ford did his best to try to assuage public fear. He got vaccinated on national TV. And this led to a lot of like, how do we prepare? How do we make sure this doesn't happen again? Drafting of policy. And since that time, right, we know viruses have continued to evolve and circulate around the planet, but we've also gained new tools, right? Our ability to sequence DNA is a lot better than it was in 1976. And we also now have these little nifty diagrams, right? We're now able to infer phylogenies. So some of the things we've learned since then, we've learned that a lot of diseases emerge from other animals and get into humans. So it's now, there's no argument, right? Emerging diseases, about 60% of diseases in humans also affect other species, and about 75% of emerging infectious diseases have zoonotic origins. We can go through kind of the laundry list of really terrifying things like Nipah virus or Marburg virus, Ebola, right? These things are zoonotic in origin. And there's a paper during the pandemic in the New England Journal of Medicine that was pointing out, look, the majority of novel disease outbreaks are a consequence of zoonosis, right? And there's real value in using evolutionary tools, right, to figure out what animals are being infected, right? What animals are infected and capable of transmitting diseases. That lets you know where you can deploy resources, right? That's really useful if you want to prepare for future outbreaks. And it's exactly what people have been doing, right? It's been some really cool work in evolutionary medicine, understanding reservoir species. I'm just highlighting some of my favorites. If you go online, I can't highlight them all. Kind of in present day, in Madagascar, for example, right, you can monitor populations of rats and figure out where bubonic plague is distributed, who has the highest concentrations of bubonic plague, make heat maps and figure out, okay, we need to really monitor over here to prevent plague outbreaks. You can do this for, we were talking about hantavirus earlier, like we can do this for hantaviruses, figure out the spread of hantaviruses due to climate change, those sorts of things. You can also look historically and figure out how bubonic plague circulated in the first place to begin to solve kind of historic transmission routes. There's been some really cool papers using evolutionary methods on that front, one in Nature that was looking at the source of Black Death across the 14th century, kind of highlighting that, oh, actually it came from this region over here, right? Using ancient DNA, they're able to extract Yersinia pestis out of digging up graves and extracting Yersinia pestis. I think you can actually extract it out of the teeth. It gets preserved, so you can actually find these DNA fragments. And so they're able to reconstruct, wow, all these ancient haplotypes are occurring from this region. And using this, right, and kind of going through these historical samples, they're able to show you have the center of origin over here in Kyrgyzstan and Tajikistan. And then you have kind of these two waves that are spreading out, one towards Turkey and heading towards Europe, the other going into China and into East Asia. And armed with this type of information, or this is something that, was also very germane and continues to be in thinking about the evolution of sort of where SARS-CoV-2 emerged from, right? There's been no shortage of papers starting right after the first sequence was published and thinking about how SARS-CoV-2 went into our population, where it's zoonosed from, what the closest sequence ancestor is. including in places like Cell, Nature, even the Journal of Medicine. And in addition to just figuring out where stuff comes from, right, we can also begin to understand how diseases circulate using evolutionary medicine approaches. Whoops. So for example, here, right, we can just look at how, for example, a disease spreads. You can kind of follow these dispersal pathways where it starts from over here, and then it kind of disperses up this way, and then you have a sort of back dispersal back to this region. So you can build these epidemiological models and actually map transition pathways. And you can also do this historically to solve some of these like big historical challenges, like how did plague circulate and continue to circulate for hundreds of years? And for that, there have been two competing hypotheses that people have argued about a whole lot. One of which is that it you have these reservoir populations in Europe. It sort of circulates at low levels and then every once in a while breaks out and escapes Europe. The other idea was that you can have multiple introductions, right? And you can have, basically you have like one source area and then you just have waves of plague one after another, and that's what's causing your outbreaks. Now, this is historic, but this would matter also for contemporary reasons, right? Anytime you think about which ways our disease is going to track, right? That's where you can put your health resources. There was a pretty cool paper in PNAS that basically showed, hey, look, if we can sequence all these ancient DNA samples, we can actually get evidence that yes, indeed, there are these multiple introductions of plague, right, that are coming from Asia going into Europe, right? So it's not this recirculation idea, it's that you have these multiple waves of introduction. And I mean, we all lived through it, right? We got to see this in real time with SARS-CoV-2, right? You could get on Gisaid anytime a new strain was announced and just kind of watch how these strains were spreading across the planet, right? Which was like really interesting to me just to see phylogeography in action, right? You can kind of see how just interconnected we were and how quickly these strains could spread across the planet. Now, another thing that was really interesting use of evolutionary medicine during the pandemic was just seeing how variants replace each other. So there's a pretty cool Nature Reviews microbiology paper kind of showing here, we can basically track in real time, right, how these variants spread, become dominant, and some other variant evolves and sort of takes over as the new dominant strain. And we're kind of watching this throughout the pandemic, right? The first mutation that was seen that was considered under selection was this D61G in the spike protein. It basically just aided in faster replication, and we saw this kind of sweep through. And then by 2021, you had multiple mutations that were arising that helped with like ACE2 affinity. It kind of kept the ACE2 receptor, kept the receptor binding domain more in an open position, so it was able to bind to ACE2 more effectively. And there've been a whole slew of other mutations, right? So we're able to track these mutations. Here's some examples from NSP6, a nucleocapsid, which helped increase transmission through basically interactions with the innate immune system and also increasing viral replication. So we can figure out where these mutations are, can make, whole heat maps here of different mutations. Some interesting ones, I'll get back to one in ORF7A that also aids in transmissibility. So including other mutations, right, that even interact with the vaccine to make the mutations, to make the strains easier, they have an easier time getting around the vaccine. And we can start building these sort of mutation landscape, these mutation maps and figuring out, okay, cool, here's these mutations, here's these mutations. We know what they do functionally. And that was like a lot of preamble just to say, this is really useful information now because now we can predict how viral strains will behave before we observe the behavior. Right? We're constantly sequencing. We've got wastewater projects all across the planet that are sequencing, tracking the emergence of new strains. And based on that, we can see what are these new strains, what mutations are there, how likely is it that we're going to see something that might go through to fixation and replace something that's there. So if we can associate general patterns of virulence and transmission with specific genetic markers, we can use that for risk assessment, right? So this is another place where evolutionary medicine can then inform policy. And kind of when we got into this, we were really curious, kind of like, like many evolutionary biologists, the first thing our minds went to was, oh, let's think about adaptive evolution. It's 2020, there's this new virus. How is zoonosis working? By the way, this slide I had in an intro to EEB class long before the pandemic as an example of cute biodiversity. And now to me, it's like one of the most terrifying images I've ever put into a talk. So it was fun to recycle and be like, oh my God. So our foray sort of into working on coronaviruses actually came from just thinking about selection. And we thought at the time, and to still do, right? If we can understand adaptive and non-adaptive processes, we can look for promising conserved targets that could be used for effective vaccine development and drug development. And in general, if you look at SARS-CoV-2, there's actually contrary to the media narrative, most of the genome is actually under very strong purifying selection, but there are some candidate sites for positive selection, particularly in the receptor binding domain, but some variance between studies. And what we did at the start of the pandemic is we just got every sequence we could get our hands on. This is again, 2020, at the time, wow, 559 genomes. Now, if you were to look at how many SARS-CoV-2 genomes were here, I don't know what the number is, because I have too many zeros for me to be able to read at a glance. There's so many genome sequenced. But we looked at the original genomes that we could get, and as well as genomes from the original SARS outbreak, as well as any host coronavirus or reservoir host coronavirus sequence that's publicly available. Did sort of our standard phylogenetic methods. Did some PAML stuff. We also reconstructed the ancestral genomes to get a sense of what was happening at that node that precedes zoonosis in both SARS-CoV-1 and SARS-CoV-2. So here's sort of a small version of the phylogeny, right? We have these bats that are sisters to this human outbreak. These humans here. Again, a clade of bats that's sister to that. And then we used a method you may or may not be familiar with called MassPuref. It's basically just a model average cluster-side Poisson random field model. So there are other Poisson random field models out there. It's similar to that, but it basically is just looking at population, without going into a whole thing. You're basically looking at polymorphism post-zoonosis and sequence divergences that were fixed pre-zoonotic transmission. To try to look at selection. So the nice thing here is it's a little bit more powerful than some of the other statistics that use these similar approaches, because it allows you to create profiles at the site level. So rather than assuming you've got selection at sort of the level of gene, you're actually able to detect signatures of selection at the individual amino acid residues. And it does this by integrating across like a whole slew of models in a very painstakingly slow way. But at the end of this, you're left with profiles of selection intensity. So here's just an example where this was used and compared against a flu data set, and everything that's in gray is where there was positive selection as identified in flu with another method. And then this model is able to basically find a few additional sites. Now we know there has been evidence for positive selection. So we did something similar for SARS-CoV-2. So basically, here's your confidence interval. If the confidence interval goes along this line, there's positive selection. If it stays below the line, it's strong purifying selection. And across most of the genome for SARS-CoV-2 and SARS-CoV-1, these genes are under just very strong purifying selection. We did find some weird stuff. So we found these sites under positive selection in ORF7A. Remember, we were doing this back in like 2020, and it was just everybody's talking about spike, and we're like, what the heck is ORF7A? Like, what is going on here? Turns out now we know. Now we know that ORF7A actually interacts with our immune system. It actually downregulates MHC class 1 expression. If you don't know what that means, picture MHC is like kind of like the windows into your cell. So if you think about your immune system sort of surveying what's going on, right, imagine that you're kind of like shuttering the windows and now it can't see it, and for lack of a better metaphor. Got it from a pop textbook about immunology, which I thought was a nice metaphor. So we find it's these same sites, and then there's also another paper that came out 2 years ago in horseshoe bats and pigs in sudden acute diarrhea syndrome virus, which is a really horrible name for a virus. Glad it's not in humans. But there you also have high sequence diversification in these similar regions of ORF7A. So there seems to be something going on here that this method was able to detect. But as we were doing all of this, we were sort of thinking about pandemic preparedness. We're sort of over in this region where evolutionary biology feels very comfortable, genome characteristics, right? We're very comfortable in that space. But we kind of drifted this way pretty abruptly. Our research program took a pretty abrupt right turn into thinking about population immunity. And during the pandemic, I'm sure you remember, there were a lot of questions about immunology that were being asked over and over and over. So, things like, can we build better can we model better vaccines? Can we make a universal COVID-19 vaccine? How often can I be infected with coronaviruses? And great questions at the start of the pandemic, like, can you get coronavirus from drinking Corona beer? Technically, there are scenarios you can imagine where this is theoretically possible. It just takes a little work on the effort, on the participant side, but yeah, it was an interesting time. But one of the big questions at the start of the pandemic was how long until someone can be reinfected, right? This was like the scary big question. And I don't know if you guys remember this. Hopefully you do. I'll never forget it. You could, on the same day, look at the news and read, scientists have just discovered lifetime immunity after prior infection. Or my patient got COVID twice, or immunity might last weeks, right? And so we were kind of all over the place between you can get reinfected quickly to you're immune for life, which is kind of the confidence interval of I have no idea. Right? It's just like sometime before you die, maybe we don't know. So this really actually matters, right? For those of you who know about epidemiology, we can think about just a simple statistic like R naught, where we're just thinking about how many people on average can a person infect. And so if we think about influenza, on average, one person can infect about 1.5 people. It's kind of like, on average, right? And then the original COVID strain, right, similar to SARS, so the average person was infecting about 3 people. By the time you get to Omicron, the average person infecting 9 people. That's a lot of transmission. And if you get reinfected quickly, that means there's a lot more transmission. If you're immune for life, that means it sort of fizzles out, right? So this actually really matters when you start thinking about, well, what do I need health resource-wise? And the challenge to solving this problem is you need a lot of data to get the answer tradition, right? You need long-term data. Right? You're going to be monitoring patients, you're going to be tracking their serum, you're going to be tracking B cells, serum antibodies, you're going to be doing more ELISAs than you've ever wanted to do in your life. You're going to be creating antibodies. Anybody who's done ELISAs is like, yeah, it's a lot of work, right? You're doing multi-year studies, tracking lots and lots of people to see how long they get until they get reinfected, and just what these average numbers might be. This is not something you can do at the start of a pandemic, and unfortunately, when you're in policy, you need that answer very quickly. Right? You need to make decisions on what the next thing is that a population needs. So you have this mismatch between the data you actually need to answer the question and the data or what a policymaker needs. So one thing we started thinking about is we were just wondering if there's another approach to get the critical information, this type of critical information faster. And the true story behind this actually is my friend Jeff and I, we saw the same newscast or variants of it on the same, we basically got on Zoom the next day venting because somebody in an interview said, we know nothing about the durability of immunity. And the words "we know nothing" got us both triggered because we're like, you have to know something, right? Coronaviruses are a thing. We know stuff about them. It's like if it just seemed like it's such an odd thing to hear as an evolutionary biologist. Like if I told you, and it's just a familiarity thing, right? If I told you tomorrow, there's a new species of pufferfish we just discovered off the coast of North Carolina. You probably immediately pictured a pufferfish because you know what a pufferfish is, maybe. But you probably didn't picture like a hammerhead shark, right? Now, when it came to talking about immunity to SARS-CoV-2, oftentimes we would talk about viruses like they were just these interchangeable fish species that are not closely related at all. Like maybe it's like measles, maybe it's like this, maybe it's like this. Like maybe it's like the other coronaviruses. So let me start there. So we started wondering about like, well, what data is out there? And then as we were working on this, just kind of give prelude as well, other things we started realizing throughout the course of this program is like, oh, we can actually start thinking about, if you have this information, you can think about vaccines as well, and think about boosting as well. And all these questions that are still being asked to the present day, except maybe some questions aren't quite so answerable by evolutionary biology. This is what happens when you Google trending COVID immune questions. Those are the things you find. But what we discovered in the course of this research is that phylogenetic comparative methods are actually really well equipped to inform the durability of immunity to an emergent virus. And at the time we started this program, right, there was no reinfection data. We started before there was documented reinfection, but Time Magazine back in March put out here's the big questions about SARS-CoV-2. One of them is if I already had, I'm trying to use a little later pointer. If I already had COVID-19, will I get it again? If I'm fully vaccinated, am I immune to COVID-19? And will I need to get COVID-19 boosters forever? These are what Time considered the big questions. And turns out you can answer all of these questions using evolutionary approaches. So the first one we looked at is if I already had COVID-19, will I get it again? So we published, I'm still really happy about this. We made The Lancet publish a comparative evolutionary study in one of their journals, and I was like, yes, the word evolution in the title. This is great. That felt like such a big win for evolutionary medicine. And what we realized is, remember, I told you guys you need all this long-term data, right? So where the hell did we get data if it's the start of a pandemic? Well, it turns out we had long-term data for these other coronaviruses. We had antibody levels and reinfection data collected almost for over 30 years from things like HCOV-229E, HCOV-NL63, HCOV-OC43. So these things that we commonly refer to as colds that people were studying, we had data from. We also actually have data from MERS, and we had data from the original SARS outbreak as well. So we had a lot of antibody data from these lineages. And we also, at the start of the pandemic, immediately people did start doing those studies and publishing initial antibody data. So we even had the start of an antibody profile. It just didn't go out very far yet. So now we can make this somewhat familiar. We can place all of this into an evolutionary framework and start to think about what do antibody declines look like following infection? What sorts of reinfection rates might we estimate? So one of the first things we did, right, is we used a technique that actually comes from paleontology, which is kind of a weird thing to do. But you guys probably all are familiar with ancestral state reconstruction, maybe? So you can look at tips of a phylogeny and reconstruct what the ancestor looked like. Well, in paleontology, they often have the reverse situation where they have a fragmented fossil and they have to reconstruct missing data. So they do what's called ancestral descendant state reconstruction. So they take these methods and they basically run them backwards to try to reconstruct what's happening in tips. So we were able to use these methods to figure out basically what do these antibody declines look like, right? So we're able to reconstruct what antibody declines look like. And from that, we have this reinfection data. We're also able to reconstruct what types of infection rates you might expect. And to me, the coolest thing is, like, now that we're in 2024, we can ask the question, well, how well did you do? Right? Which is, the big question, right? It's like, sure, you can use evolutionary methods, but do they work? So empirical studies, right? So here's one example. Here's an empirical study that came out like a year or so after our study, where now we had infection data 420 to like over a year after COVID first emerged. And so unvaccinated individuals, you get about a 34% reinfection rate around 420 to 480 days. Our estimate was 34% probability of reinfection at 450 days. That's pretty good. So another validation by an empirical study. Here's another estimate. They just use a different way of looking at this, but another study that came out a year later. They're saying about 18% reinfection around 275 to 300 days. We got 18% probability of reinfection at 270 days. So this worked, right? And it was able to get answers, 270, more than 270 days earlier, right? So just by having this information on these close evolutionary relatives, we're able to fill in this gap. Now, as we were doing this, remember this, we were getting this data kind of before there was any reinfection data. And now we were at a point where vaccines were coming online. And the next question was, how long are you immune after these vaccines, or after you get a vaccine? And so what we did is we got data from Oxford AstraZeneca, Pfizer Pfizer's vaccine, Moderna's vaccine, and the Johnson & Johnson vaccine. And we were able to do a similar sort of analysis for these 4 vaccines. And turns out there was some really nice data relating the Pfizer vaccine to antibody levels with natural infection. And then what that allowed us to do is also start thinking about boosting and doing an experiment in simulation, right, where you can, not really simulation, but you can quantify, right, over the course of 6 years, what are infection risks if you boost at different intervals without having to do a 6-year study. So kind of, when we think about this, right, if you're thinking about antibody levels over time, we can kind of just consider, we can think about, well, what happens if, following vaccination, your antibody levels are higher versus your antibody levels are lower than that of natural infection. Obviously, it's going to lead to having a longer duration of antibodies versus a shorter duration of antibodies. And then you can do the same sort of exercise, right? You can relate this back to a probabilistic model and thinking about breakthrough infection over time. And again, if your antibody levels are higher versus lower, right, that leads to a longer or shorter durability of immunity. And so we did this for all of these vaccines using data from Pfizer and Moderna, Johnson & Johnson, and Oxford AstraZeneca to basically figure out what the durability was of these different vaccines. A couple of things, we're kind of limited in the data we can use. So we have to sample near peak antibody levels to make sure we had COVID naive subjects, as that gets harder and harder as the pandemic goes on. Proper vaccination timing had to be present in data. They had to have a comparison to the Pfizer data because that's what we could compare our data to. I forgot to put this on here. Also, consistent ELISA assays is really important for those of you who are immunologists. Jeff's shaking his head like, yes, it is. some of the variance between those is actually enormous. And there's been some really interesting papers on some of the early ELISA kits that sort of disappeared, and you understand why they disappeared. But what we found, first, of course, here's the natural infection. The Moderna and Pfizer vaccines, the mRNA vaccines actually produced a much higher signature or a much higher antibody response to natural infection, whereas the two viral vector vaccines produced response to vaccination that was much closer to natural infection, if not slightly below it for the Johnson & Johnson vaccine, which kind of mirrors the efficacy. So what we were able to do with that is we were able to say, we were able to go through and just look at, like how, what the median times were that you'd expect to break through infections for the different vaccines. And again, we can do that thought exercise now of how well did we do with these antibody profiles. Well, here's a data set that came out a year later at the end of last year, and they got, they were tracking, I think it was Pfizer's vaccine, and they were just tracking over close to 300 days. So the antibody level from peak is at about 12% reduction by 270 to 300 days. We were at 0.85, so about 15% reduction by 270 days. So again, we're pretty close to the empirical estimate. So, these methods are like, there's not that much actual, part of the reason is there's not much of a variance in the coronaviruses and their immune response in general, outside of MERS is a little funky. So we're able to get these estimates that work really well for these types of problems. So the next thing that we did is we started going through and wondering, okay, so when should we be boosting? What's an appropriate booster interval that we should start thinking about? And to get at this, we looked at Pfizer and Moderna because they had very similar antibody responses. And we just looked at what happens if we boost every 6 months all the way up to 3 years. What does that do for infection risks over a 6-year period? And if you don't boost at all, and you just go for 6 years, you'd expect about 9 out of 10 people in your population on average to get infected. If you're boosting once in the middle there, about 75, 77%, you'd expect to get reinfected. If you're boosting every 2 years, about 64% reinfection rates. By the time you get to 1.5 years, about half your population you would expect to see get reinfected. And by the time you get to 1 year, this drops down a fair bit, right, down to 31%. So it's a very big difference, right? 3 out of 10 versus 9 out of 10 to me is a very big difference in numbers, especially if you're thinking about our health resources, right? So this is really interesting to know. And then this is actually kind of amazing, right? If you were to boost every 6 months and kind of keep up with the virus, you'd be down to about 10% of your population. So this was, I thought, really cool work. We were able to actually show this to FDA, which is really interesting to me as a fish biologist. Was kind of one of those terrifying moments where you never felt. I feel so much more comfortable working underwater around sharks than I do in a room full of people deciding public health policy virtually. I'm like, I am not trained for this. This is amazing. It was a really cool experience though. But what we found, right, is we found what's now been kind of validated, right? The mRNA vaccines at the time were producing a longer duration compared to natural infection alone. Caveat here, as vaccines change and as viruses evolve, it doesn't always mean that there's something intrinsically special about the technology, right? You have to have the matching antigen profile for the vaccine to work well. So it's not necessarily the technology alone. It was just they did a very good job modeling what they're putting in their vaccines. And also there's a real delay, there's a cost, right, to delaying booster administration. And that has a consequence in terms of the amount of infection that you would expect. And so this landed on the yearly to bi-yearly population-wide boosting as a good decision. Now, a big caveat to all of this work, right, is we're assuming kind of the average of averages, right? It's like the average population of the average is the average. We're not taking any, immunocompromised individuals, people who are pregnant, people who are younger, people who are older. We're just taking the average of humans. So immune state really matters to these sorts of projections. And one thing that we became interested in is as we were getting new data is looking at other populations, in particular because the FDA last year, I guess a year and a half ago, I can't keep track of time. In the not too long ago past, I'll hedge my bets, they released updated guidance for healthcare practitioners saying, for patients who are immunocompromised at the discretion of your healthcare provider, you can boost those individuals again, which is great. That's absolutely great. The problem is how much? Right? There's not really much guidance there other than, yeah, you should probably get another booster, but when? So one area where we started looking, there's a lot of data on people on anti-cancer drugs. So we have data on healthy controls, data for people on targeted and hormonal therapy, people who aren't undergoing any therapy, people post-chemo, people who are on immunotherapy after chemotherapy, patients under just immunotherapy, patients under stem cell treatments, as well as patients on a therapy called rituximab, which you may or may not heard of. I'll tell you guys about that in a second. So we're able to do similar types of analyses and just look at what happens when you're on these different therapies for shorter durations of boosting. So here, just if you're immune typical over a course of 2 years, we're doing shorter term here, but over the course of 2 years, you have about a 30% breakthrough risk if you're just not boosting versus about a 4% risk if you're boosting every 6 months. And across things like targeted hormonal therapy, stem cell therapy, immunotherapy, and immunotherapy, I don't know what just happened. I'm clicking buttons too fast in too many directions and the computer can't keep up. Animations. Anyway, once the animations catch up, these are all very similar. So we have about similar risks, right, for these different therapies. So the guidance that holds for the general population is very similar for these populations of patients. As well as chemo, having immunotherapy after chemotherapy also kind of falls into this category. But there are some differences, right? When you get into patients who are just on chemo, infection risks do go up, as well as for patients that there's data on so far that aren't undergoing any treatments. Again, the risks are going up. So alternate booster schedules might be warranted. And again, we're not healthcare providers, we're just giving risks. The thing that really stuck out though is this therapy called rituximab. Rituximab is a very interesting therapy. At the start of the pandemic, this was the thing that practitioners were absolutely terrified with because it wasn't clear if even after vaccinating you can mount an immune response that destroys your B cells because it's trying to keep cancer from spreading throughout your bloodstream. Basically, there was a bunch of papers that came out and said, okay, cool, you can at least mount an immune response, but even after mounting an immune response, even boosting every month, I think is what this one was, or every 2 months, the risk of infection just still is very, very high for that amount of boosting. So not surprising given what this therapy is doing, but good to get some numbers on if you're a practitioner. And we were doing this in conjunction with somebody who is an active clinician in this arena who is very interested in these results. Since then, we've also been working in a lot of other high-risk populations. So another paper up on medRxiv now looking at MS patients, if you're interested in that. One of the things I wanted to point out is we have data on another therapy called teriflunomide. This thing does do things to your immune response, but there's a nice discussion in here about the other thing that these models can't do, which is think about the therapy itself. If you're up on your COVID therapies, you'll know this actually is a COVID therapy. It interacts with the virus itself. So we have a little discussion in there about thinking about the limitations of these models if you're interested. We did have a limited set of rituximab data. Rituximab is also given off-label to MS patients. Unfortunately, the data that we could find, the antibody levels actually get so low in those patients, they go below natural baseline, which these models can't handle. We get into like weird mathematical areas. So basically, it's really low, like you would expect. So it's the same sort of caution. We just can't actually put numbers on it because we assume that there's some baseline that you're returning to. And there's only like 2 patients and the numbers were just too low to get a good reading on. Now, the other thing that we've become really interested in is thinking about the elephant in the room that's left is how many of you have had COVID? I shouldn't ask personal health questions. Think in your head, because I don't actually want to know the answer. I'll tell you about myself. I am amazing at getting COVID. I can schedule a vaccine on a calendar and get COVID like 3 days before I'm going to get my vaccine after a faculty meeting. This literally just happened. So, it's like, breakthrough infections have been the bane of my existence, which is ironic because I like doing all this work on vaccination. They're like, so you got your vaccine? It's like, no. Because COVID finds me first. It knows where I live. My wife teaches in preschool, so we're constantly exposed. And the vaccines have an unfortunate rollout date of when we start to get into these fall surges. But the question becomes of what happens if you just got an infection? Now when do you boost? And currently the guidance is, you can get 3 months. Sure. And Yeah, as I'll show you guys, that works sometimes, but if I were to stop that clock and stand here for a while, in 12 hours, it'll be the right time again, right? So it works sometimes, but there might be situations where that might not work so well, depending on what's happening in the landscape. And what we're about to see again, we see this every year, like it's a big surprise. It's flu and cold season, and there's a lot of COVID, right? And this was the headline from last year in January. I'm willing to bet money right now we'll see headlines like this again in January. So we know that there are these surges in our population, right, where we have higher incidences of coronavirus and lower incidences of coronavirus in other parts of the year. So right now, our models have been assuming there's a kind of this steady state, right? Like it's just you can get COVID anytime you go anywhere, right, at an equal rate. But we know that there are some times when there are a lot more people infected, which means that you have a higher probability of getting somebody transmitting to you, right? So this kind of leaves us in this confusing state space if we're thinking about what to do, right? Well, we have to weigh in, thinking about what's going on in the population, weigh in, thinking about our own immune health status, which might be different from our friends, which might be different from the other person we were just talking to, and trying to figure out, well, when do we expect that we should get the next vaccine boost? And turns out you can answer this question using evolutionary methods too. Now, we did this kind of in a curious way. So one thing we've been curious about is kind of, and this one I can't tell you if we're right yet, by the way. So I'm just going to tell you we did it, then we're going to use this data, but you can use any data you want for the incidence, including like what you might expect for next year. We did it under endemicity. So we were just curious, what might endemic COVID look like once we sort of get into like the true endemic state? We're sort of in this weird we're somewhere in between pandemic and endemic. I don't really know, right? But we have like different population statuses. We still have the aftermath of heterogeneous like health interventions across the planet. So we're wondering, okay, if you give us a few decades, for example, what sorts of seasonal patterns might we get? So we had a paper out in MBio just doing this for different parts of the world, where again, we're just projecting what we might expect SARS-CoV-2 incidence patterns to look like based on the incident patterns of other coronaviruses in similar geographic areas, which is basically just a big meta-analysis of flu and cold season, where you're like, yep, this is what Denver's flu and cold season looks like. That's what New York's look like. So if we were to assume, though, that you have this sort of general pattern, one thing that we can do is we can start to think about how does that change the probability of infection, given you're infected on one day and you're just kind of like trailing throughout the year. And the thing just to point out here is you get these kind of like, valleys, right, where your probability of infection is low, and then you have other times where your probability of infection is higher. And based on this, we can actually estimate, well, what would be, if we assume like an incidence pattern that's consistent year to year or roughly consistent, again, there's going to be variance around every flu and cold season, right? We can just see, okay, how does that change over time when you're not boosting? How does that change your probability of infection? And how do we find an optimal date that we can boost on to keep the cumulative probability of infection at its lowest point? So based on this sort of analysis, we can actually determine, here's kind of the date for New York around, like actually for New York's around September. Mid-September is a good time to probably boost your population right before the onset of the big fall surge. And there are other parts of the world where that's not so great of a date. Now, if we have that though, right, we can also do one more thing. We can think about, well, what about for an individual that's infected on any given day of the year? When should you boost? And we can actually create these really cool kind of probabilistic maps where we can look through infection across the year and actually figure out, okay, cool, I was infected in June, when might I want to boost next? Oh, okay, I can actually wait like a whole year and go on until like November or something. So depending on when you're infected actually has a really big consequence to when you should get boosted next. So sometimes, yeah, if you're infected at a time scale, there's some, like if you assume you're going to get boosted sometime in September, for example, then yeah, there's a time when, yeah, within getting infected, a couple months later is pretty good, but then other times where you can wait actually a pretty long period of time before your next booster shot. So, and I'm pretty excited about this because it kind of provides an answer for at least thinking about when is a good time to boost. So I'm going to minimize my chances of being sick and not get COVID for the 4th year in a row. But kind of, we have that up on, actually that might be coming out in clinical infectious diseases soon. We're doing some other really cool stuff, which I'm not gonna have time to go into, but one of the things we're really interested in is actually starting to think more about variant evolution and antigenic evolution. So one of the issues we have that was also brought up with the FDA, right, is we're manufacturing something, but it takes a long time to go from the design to the deployment of a product. Meanwhile, the viruses are continuing to evolve, right? So depending on how good of a target you have, right, that's going to change the efficacy of your vaccine. So we're doing a few things, thinking about sort of vaccine rollout and modeling the consequences of that. The thing that I'm also really excited about is thinking about how vaccines are actually shaping variant evolution. There was just a paper out in Cell showing this as well, showing that, hey, when you do these like pulses of vaccination, the viruses respond. Shocking, right? Think about it, it's like, yeah, of course they will. So sort of thinking about, the rate of antigenic evolution, the effect of local vaccination drives, and sort of thinking about how that shapes viral evolution, and then thinking about how that goes back into, the natural decline of antibodies across a population to thinking about immunity. So it's been kind of like a really cool landscape I've sort of stumbled my way into over the last couple of years. But in conclusion, right, just kind of wrap this up. I think the neatest thing to me about all of this is really just seeing phylogenetic comparative methods being used in this way, and thinking about how we can think about phylogenetics as part of the toolkit for pandemic preparedness and pandemic response. And in our case, I'm just showing ancestral descendants data analysis and how you can use that, right? You have a new emergent disease. Let's fill in some unknown parameters. And if we were just to right now collect all this baseline data on clades that have pandemic potential, right? Imagine what we could do next time that there's a pandemic, hopefully not, but if there will be, right, we would have all this data we can then use to go, hey, immunity isn't between lifelong and 3 days, right? It's about this, right? Or some other aspect of a disease. So to me, I think, there's tremendous value in just collecting this type of data now and being prepared. So we do some grants in to try to do that. Shocking, coming from a museum, I view all these historic blood samples that have been sitting in people's freezers, especially the controls for other studies, as like a gold mine for this. There are a lot of samples out there. We don't need to start from scratch. There are decades and decades of blood bank samples we could look at right now to do this kind of work. But myself aside, I think to me, one of the big lessons too, kind of bring us full circle, is throughout the entire pandemic, right, we really saw evolutionary medicine step up and really provide a lot of answers. So, I'm highlighting this, but you could literally re-give this talk and highlight so many different things that people had done, right? Thinking about tracing how the diseases spread, tracing mutations, et cetera, et cetera, et cetera, right? Evolutionary medicine was like, we saw phylogenies in the New York Times, right? Like, it was amazing just to see, okay, we went from sort of this historic science to a science that's front and center and providing context and information. So I just want to thank my team. It's been just really awesome getting to work with everybody. So Jeff Townsend and I, we've been on Zoom all the time. This is his wife, Allison. Nancy Ruddle, if you guys don't know her, she's an immunologist at Yale, who's probably like the most patient person in the world. So Jeff and I have a lot of ideas sometimes, and Nancy is the one who's like, that's not how it works, guys. So she spent a lot of time literally going through everything to kind of get us up and running. Sadir is great. We do a lot of molecular clock stuff. Haley did a lot of the, Jeff and Alex have an idea who's going to actually do the thing. That's Haley. And then also Brenda is a clinician MD who did a lot of who's really been involved in thinking about the different patient subsets. So thank you guys very much for listening and inviting me. And happy to get started. That clock is fast, but it made me really nervous. Yeah.
So I'm not sure if that's true with COVID virus, but it did show that over time, the virus became more infectious, yet supposedly less lethal. So there's some sort of attenuation happening. Can evolutionary medicine methods explain that?
There's oh my gosh, there's some really interesting talks on this that have wondered the same thing. I saw a couple talks on this, because has it become less lethal, or has the population been previously exposed? So it's less lethal in those that have already been exposed and have some protection against the virus. So that's actually an area of really active debate. And early on, I think there was a lot of prediction that it was just going to trend towards being more benign. That the big question is, if we were to find, this is a horrible experiment, but if we were to find a population that's completely naive, would it still have the same mortality rate or not? I don't know. And there is some evidence suggesting it hasn't become less lethal. It's just that a lot of the population that was highly susceptible, not the best outcome occurred. And now we're left with sort of a population that's no longer immune naive. So we have some degree of protection. I will say a lot of these viruses, even there's cross susceptibility, or like cross susceptibility, that's the word I'm trying to use. if you have antibodies for one of these coronaviruses, they'll often interact with, they'll also, so that this actually becomes an issue in ELISAs as well when you're doing these sort of historic studies. So you're like trying to tease out which one of these is it, because this ELISA seems to be detecting both.
When you're making these predictions of the risk of reinfection, are your models currently taking into account the Unix evolution in the viral population?
No, we have some math to finally do it. But we haven't yet. So right now we're sort of, well, technically we are, right? Because the baseline data, this is the beautiful thing about this data that's kind of horrible at the same time. We never once tried to intervene over the last 100 years against any of these other coronaviruses. So by default, the infection data has antigenic evolution baked in, right? Because we never did any health interventions. So it's sort of the natural pace of evolution. So technically, yes, but now that we are dealing with, now the big difference is we're vaccinating. And so we are now thinking about, okay, how do we actually tease apart antigenic evolution for this particular virus? Yeah, but that was kind of, when we first started it, we realized that's the only reason this works, right? It's because, yeah, we've never, right, we just get colds and go to work is kind of was the model. Yeah.
I was interested in something you said at the end there, thinking about how the vaccines are pushing viral evolution. Is anyone working on sort of this cycle of, can we have a virus that pushes viral evolution in a way that makes it more susceptible to next year's vaccine?
Oh, that's so predicting. So the idea would be that you're trying to predict where the virus will trend to have a vaccine that's going to head it off, kind of.
Yeah.
I haven't seen that for vaccines, but that's an idea in cancer. So the idea there is that you know kind of what mutations will arise given a slew of therapies. So you try to design something where you're basically you're trying to channel the evolution of the cells in a way where they reach a dead end. Is one of the evolutionary ideas that I think a lot of people over at Duke are looking at, like Jason Samarelli's group. But I haven't seen it for vaccines.
Have you had the chance to look at, like, for example, coronaviruses in like a mammal that has not jumped to humans yet to maybe possibly predict kind of the jump to humans even before it happens?
So yeah, that's something. So there's a couple things that I want to do along those lines. So one thing is actually the reverse of that. So we've been infecting all of these animals in our landscape. So one thing we could look at is like what's happening in these white-tailed deer populations, for example, where we're getting kind of transition into zoonosis in reverse. In the coronaviruses, I had a student who really wanted to do this and got me really excited. So we realized there's no data, right? So there are even coronaviruses in, actually, there's a vet school over there. There are coronaviruses in cats and dogs. Somebody should collect that data, right? Because, yeah, there have been, throughout the pandemic, they were very cryptic. But every once in a while, a paper would come out where people were going into freezers. And I just remember one in Indonesia that stuck out to me. One of the ones I mentioned was NL63. NL63 is an odd coronavirus that tends to infect children more, especially very young children. And there was a village, I think it was in Malaysia, that had a high outbreak of pneumonia. And they actually went through the freezer samples to see what caused it. Turns out it was a coronavirus closely related to NL63, infecting the same target population, right, children, and they actually identified it came from a dog. So the data might literally be like a few blocks away to do exactly that. But yeah, we couldn't find anything in the literature. There's nothing really, like there isn't enough out there. Maybe now, we looked a couple years ago, there's been a lot more interesting coronaviruses.
One more time for one more question.
So with the breakthrough studies, so there's conflicting data whether or not coronaviruses or the SARS-2 is following like influenza with the original sin, and the idea that what you first got vaccinated for, your immune system is predisposed to that serotype. Can you see that in your data, that whether or not vaccine is actually skewing the human population to respond to old variants while the new variants move off in different directions?
So the Cell paper that just came out was talking about exactly that. Where they were showing some evidence that it's possible that basically you're getting a response of these variants to a large subset of the population being vaccinated and selecting for higher immune evasion of vaccinated individuals. Yeah, so that's the first time. It was kind of like this thing. We were just like, yeah, it'd be cool to look. And then this paper literally came out like within the last month, or maybe even last 2 weeks, like very, very recently, showing this for the first time. So I think that's a new frontier in thinking of, I don't know, we've just, never really looked until now.
So I just want to let you know, if you want to chat some more with Alex, he's going to be hanging out here for about like the next 20 minutes. So if you have more questions, come on up and you can ask him questions. And everybody else, thank you for coming, and let's thank Alex one more time.